I'm Matt,
I'm Gretchen. I'm Carmela.
I'm Logan.
and we did Klebsiella pneumonia.
Yeah, it's probably better.
So microbe identification, the scientific name being Klebsiella pneumoniae. Common name is just K. pneumoniae, because it's a common cause of pneumonia. It can be classified in the domain bacteria, phylum Proteobacteria, class Gamma Proteobacteria, order Enterobacterales, family Enterobacteriaceae, genus Klebsiella, species pneumonia. So its morphology and characteristics, it's a gram-negative bacillus-shaped bacteria. It's also encapsulated with a polysaccharide capsule, which aids in its virulence factors. It's a facultative anaerobe, which means it can grow in anaerobic and aerobic environments. It's also non-motile, and it has a lipopolysaccharide on the outer membrane, which also contributes to its virulence factors and its ability to evade the immune system. So here we can see a diagram of the human body, the classical and hypervirulent Klebsiella pneumoniae. Common is UTIs and respiratory infections. Hypervirulent is more causes problems in the brain like meningitis, problems in the muscles, and so the habitat and the environment niche. Its natural habitat is soil, water, plants, and when it gets into the human body, it's the gastrointestinal tracts of humans and animals. It's commonly present in hospital environments, and it's one of the #1 hospital-acquired infections. It also thrives in moist environments, so the respiratory system, urinary tract, all that stuff. And it can survive on lots of medical equipment, so like catheters, that kind of thing.
What's going on over here?
What's going on over here is the pathway that it's transmitted. So people will pick it up on hospital surfaces, and then they touch their face, it gets in their mouth, into their stomach, or they breathe it in, into their respiratory system.
Real bad.
That's where it colonizes.
Digestive too.
Yep, digestive system too, and then they shed it, and the cycle repeats. Here we can also see some classic. Okay, perfect. Here we go. Here we can see some common diseases it can cause: meningitis, endocrinitis, problems with the eyeballs. The classical ones being UTIs, pneumonia.
Liver abscesses.
Yeah, liver abscess.
Splenic abscesses, that's crazy.
Yeah, very nasty. So the growth and metabolic requirements, its nutritional requirements, it is able to ferment glucose and lactose, and it produces acid. It's facultatively anaerobic, like I said before, so it can perform both aerobic respiration and fermentation in anaerobic environments. Optimal growth, it can grow in MacConkey agar, ferments lactose where it turns pink, and blood agar. And it grows between 35 and 37 degrees Celsius, which is human body temperature, and it thrives best in neutral pH. Here we can see a diagram of the bacteria. Here's the polysaccharide capsule. Here's it getting into the lungs, and yeah, it's gonna cause a bad day.
Not gonna have too much fun with this one.
No, not at all.
Okay, so medical relevance. As said before, it can be contracted in a hospital setting, and this is a major cause of hospital-acquired infections, which that relevance to medical. And then it is CRKP. I can try to say that, Dr. Brace, but we know it's not going to end well. And it does possess a significant threat to treatment because of that resistance. And just our little pictures here, like doctor, hospital, and that. Okay, so pathosymptoms. So, Matt said before, for classical conditions, it can cause pneumonia, bloodstream infections, like leading to sepsis, urinary tract infections, so known as like our UTIs, wound and soft tissue infections as well, and some symptoms of each of these. So pneumonia, fever, chills, chest pain, difficulty breathing, and then some symptoms of a UTI, painful urination, and even blood in the urine. That would be a little more severe. Then sepsis symptoms include low blood pressure, and organ failure, not good
What's going on
at
over here?
all. Then in these pictures, on your upper right, this is a x-ray of the lungs, and it's demonstrating, it's a little hard to see on here just because the contrast. The right lung is affected more with this bacterium causing pneumonia, and this is why it would be kind of hard to breathe, because obviously if your lungs are all clogged up, then it's going to be hard to breathe, you're going to be coughing and not feeling great. Then on this picture below, sorry, it has a UTI diagram, and it's just showing how your kidneys are connected obviously to your bladder, and how this gets affected and could cause inflammation and then infection and that pain of going through your urethra. Yeah, next slide.
Ooh, I got this one. All right, so we got some mechanisms of action here. It's a polysaccharide capsule, prevents phagocytosis. It produces an extended spectrum. I'm not even gonna try and say that because I'm gonna get it wrong. Basically, it leads to antibiotic resistance. It'll form a biofilm on medical devices like catheters. Really hard to get rid of, really bad for the body. Definitely don't want that. Symbiotic relationships, it's an opportunistic pathogen, and it's usually harmless in the gut, but it's dangerous when introduced into the bloodstream or the lungs. It's pretty much a diagram what's going on. Yeah, not
All
very-
the cells are kind of being attacked and trying to do their job against this bacterium.
Yeah, it's a pretty gnarly bacteria though. It's not clean pretty easy. Alright, so historical relevance. It was discovered in 1882 by Carl Friedlander. Fun fact about him, he died from this. Pneumonia got him. It definitely got the best of him and he died. He isolated it from the lungs of those who had died from pneumonia. Noticed it had a capsule on it. Pretty crazy. It was originally named Friedlander's bacillus until 1886, where it was given the name, oh, you could try and say it.
And now you want me to spell pneumonia?
Yeah,
Oh,
no, we're not
gosh.
too good at pronouncing it.
Yeah.
Scientific wise, pneumonia is considered the most common cause of hospital-acquired pneumonia in the U.S. today. It accounts for 3 to 8% of all, oh god, nosocomial bacteria infections in the U.S. It's often multi-drug resistant and has been studied for its susceptibility to various plant extracts. It's significant in medical research and infection control. Antimicrobial resistance helped develop new technologies aimed at rapidly detecting AMR.
Yeah, and also because of that hospital or it being on medical instruments, like this plays a role in why it's so common in the hospitals today, especially in the United States, because it's so hard to get rid of.
I think a lot of people have pneumonia.
Yeah. And there's so many different like severities of pneumonia.
Alright, so we got some control methods here. Isolation, just washing your hands, sanitation, and cleaning. I feel like that's a lot of microbes. It's definitely big
Just
on
proper
washing.
hygiene.
Yeah, just be clean. It's pretty much all it's asking for. Regular surveillance of resistance, just got to keep an eye on it, make sure it's not doing anything crazy, it's not getting any worse. Rational use of antibiotics, if you get it, there's ways to treat it. Effective infection control measures needed to be strengthened to slow down the production of multi-drug resistant bacteria and control their spread. Yeah, just try and control it. Don't let it get out of hand.
And then Also, in the key word here is the rational use of antibiotics because of that antibiotic resistance, which was kind of talked about on the slide before. And then this goes to cultivation methods. So pneumonia is gram-negative, as said before, and it could be grown on MAC plates, EMB, blood agar's, like we talked about, and it's optional. Growing is 37 degrees Celsius, normal, usual human body temperature, and it should be incubated for 24 to 48 hours. And again, it's a facilitative anaerobe, so it can be grown in both aerobic and anaerobic environments. And then enteropleury test can help ID K pneumoniae.
We've done those before.
Yeah, and we do those in class, so it was nice to see we could see that connection.
So basically, the ethical impact here, antibiotic resistance. There's just multi-drug resistant strains. It makes it really hard to treat. It's pretty dangerous. Ethical concerns and overuse of antibiotics in hospitals and agriculture. Yeah.
And then the healthcare burden of this, longer hospital stays, need for more expensive last line antibiotics. And with this antibiotic resistance, more and more antibiotics have to be made. And then that's more money going into them. And this then increases time, and time is money.
It's also
So,
just super dangerous. I mean, my friend's dad who had cancer was in the hospital. He got pneumonia, and that was...
I mean, yeah, my dad was in the hospital for 2 months with a chest tube and from his pneumonia, so. Said it was worse than his stroke, actually.
So this is basically a summary slide of what we got going on. Again, it's gram-negative. It's encapsulated. It's facultatively an anaerobe. Just go bang
And
from there.
then, habitat, it's found in soil, plants, water, GI tract, respiratory tract, urinary tract. That's neutral pH is 6.5 to 7.5, and it thrives in 37 degrees Celsius environment.
It's a major cause in hospitalizations. It grows, or we can see it on an MAC plate. It fertilizes lactose. It spreads through direct, indirect contact, causes pneumonia, DTI, sepsis. Said this all.
Those are the most classical ones. And then the mechanism of it, the action is the polysaccharide capsule produces these biofilms. Then the ESBLs and carbapenemesis increases that antibiotic resistance. And thankful to our man Carl
What's that?
Freelander, it was crucial to infection control, and he's the one who found this. And he was understanding the antimicrobial resistance, but sadly, he was taken by it, which is just like crazy.
It's
like
a crazy
the bacteria you're studying. I wonder if it's when he was studying it, that's how he got it.
I wonder if he knew he had it though.
Yeah.
So basically, I mean, the ways to control this, like we said before, be clean, be sanitary, hand washing, basic PPE, don't overuse antibiotics, just be sanitary. That's all you gotta do.
Then same thing, cultivation, like we talked about, and ethical sociocidal impact, multi-drug resistance, making it difficult to treat. Doctors may overuse antibiotics because it's easier just to write that than actually figure out what's going wrong, and causing longer hospital stays, more money, more time for patients as well.
Not good all around.
And then this is our citation slide.
Pretty fancy. Pretty
Yeah,
nice.
and then Gretchen was doing our point, tap.
She did an
thing.
amazing job.
Yes.